Hi everyone, thanks for checking out this video. So this is going to be a quick one, I think, basically just showing you guys how to download the data set that we're going to be using for the second project, and we'll open up the data set and take a look at it basically. So just to remind you guys, this is a fake data set. It is just like a mock data set with fake data that I made up. The reason for that is that unlike in the first project where we could actually get like a real gene expression data set on like a government repository website, with this type of data, like people's DNA sequence data, there's a lot of like privacy concerns. So it's more difficult to find it like publicly available, basically without needing to go through like a formal like data request process, basically. So yeah, I basically made up like a fake data set that will basically give us like similar information to what a real data set would give us, and let us do like somewhat of like a realistic analysis. But yeah, it's using like a mock data set that I made up basically. So yeah, I'll put a link in the description for where to find this too. But just to show you guys, just go ahead and go to my GitHub page. It's github.com / and then just my name, Mike Saint-Antoine. So yeah, go ahead and go to my GitHub page and then go ahead and click on Comp Bio Tutorials. And then one option here is like this is the repo where I put all of my YouTube tutorial code. So one option is you can just clone this whole thing if you have some experience with Git and you know how to like clone from the command line, or you can just grab it as like a zip file here. And unfortunately though, and like sorry about the inconvenience with this, but if you do that, that will give you this whole thing, which is probably way more than you want for the purposes of this series. So if you want to just get the data set and the code we're going to be using like for this project too, you can go ahead and click Intro to Bioinformatics, and then Project 2. And then this folder here, Project 2, basically has, first of all, before I show you guys the data, it has our like actual code if you guys want to follow along with the code. So this one here, genomics.py, is the raw Python code. So I tried to put a little bit of like yeah, like comments and stuff where I could, although probably to be honest, it's not commented as well as I could have done. But yeah, it's got a little bit of comments, but mostly just the raw Python code. So one option is you can follow along with that. So if you want to download this like regular .py Python file, you can go ahead to these three dots and then just click download. The other option, which I think might be a little bit more informative, is the Jupyter Notebook file. So this file, the Jupyter Notebook, also has the exact same code as the .py file, except this file has more of like an actual explanation of everything. I tried to write like an explanation of what every part was doing. So even with this file, like maybe you could just learn from this without even needing the videos, to be honest. Maybe like I did try to write it clearly enough that someone would hopefully be able to just like read all this text I've written and like make sense of it without even needing to watch my videos even. So that's kind of the idea. But yeah, it has the same Python code as the other one, but it's got much more of like the actual like explanation of what all the parts are doing. So yeah, that one, same thing. If you want to use the Jupyter Notebook, you can go ahead and go to the three dots up here and then click download and go ahead and download it. And then lastly, yeah, there's the actual data. So if you go in this folder data, it's our example data.csv. You can go ahead and click that. It won't be able to show you a preview because it's like too big of a file basically. But you can go ahead and click the three dots and then go ahead and click download. So yeah, that should go ahead and download it. So you should have it then right over here. And I'm going to then go ahead and open this up. So we can take a look at it. And yeah, it is a CSV format. So like it is like a file with comma separated values. But if you have Microsoft Excel, you should be able to open it up like pretty easily like that. Although maybe I'll also show you guys just so that you can see that you can also open it up with like a text editor too if you don't have Microsoft Excel. Then it'll just look like this with the actual items being separated by commas basically. But yeah, I think it's a little bit easier to look at in Excel just because it looks more like a spreadsheet that way. But yeah, so this is basically just like what we were looking at on the slides before, in terms of the format of the data, but just much more of it. We now have like the actual 48,000 or so SNPs that we're going to be looking at. And then we have the 200 samples where 100 of them are normal people, and then the other 100 are zombies. So each column is a sample in one of these 200 people we have. And then, yeah, just like we were talking about before, each row is a different SNP. And then, yeah, again, at this point, just repeating stuff that hopefully you guys already know from the previous video. But yeah, for each SNP, the first 3 columns basically tell us some info about the SNP. So we have our first column telling us the SNP position in terms of both the chromosome number, where remember they go 1 to 22, and then the sex chromosomes X and Y. So we have our chromosome number, and then the coordinate of the actual letter that we're talking about. And this coordinate here is on the reference genome, so telling us what coordinate the SNP is on the reference genome that all of these people were are being compared in reference to, basically. So then for this SNP position, we have our reference allele, and then we have oh, actually, yeah, this must be, I forgot to edit this actually. This should say alt. Yeah, just have it matching the slide. I think I was calling it like the var for variant allele before, but now I think actually yeah, calling it alt is what you usually see. So I'm gonna actually make that edit now, and then I'll upload this again just to have it be changed up on GitHub too. So yeah, just to have it match the slides, change that to alt rather than var, but it's the same idea. We have our reference letter, our reference allele that could be in this SNP position, and then we have our alternate allele. So here it's a T for the reference and a C for the alternate. And then again, just like we talked about on the slides, hopefully this is all just repeating stuff that you guys already know. But then these 0s, 1s, and 2s basically tell us for each person, how many copies of the alternate allele do they have in this SNP position? So if you guys remember, we have 2 chromosomes. So for each one of these chromosome coordinates, we have two copies. So for each person in this chromosome position, they have 2 copies of this chromosome. So maybe in this position, both of their chromosomes have a T, or maybe both of them have a C, or maybe one of them has a T and the other one has a C. So yeah, it's two copies, and they could have the 2 copies of the reference, 2 copies of the alternate, or 1 of each. And again, a simplifying assumption I made when I made up this data set is that for each SNP, there's only 1 possible alternate allele. That's actually, again, not the case in real life. In real life, you could have some SNPs that have more than 1 alternate allele, basically. But yeah, for our purposes here, if someone has a 2, that means they have 2 copies of the alternate allele, in this case a C, in this chromosome position. If they have a 1, that means they have 1 copy of the alternate allele, which, since there's only 1 possible alternate allele, that means their other copy must be the reference allele. So this person here, this third normal person in this SNP position, we know that one of their chromosomes in this position has a C and the other one has a T, because they have a 1 here, which means they have 1 copy of the alternate allele, which means their other copy must be the reference allele. And then a 0 means that this person has 0 copies of the alternate allele, which means that both of their copies must be the reference allele. So yeah, again, that's just repeating something from the previous lesson that hopefully already makes sense to you guys. But again, just if that is confusing to anybody, just let me know in the comments and I'll try to further explain, because I realized this encoding of this information in like zeros, ones, and twos might be a little bit confusing at first. But yeah, basically just telling us for each SNP position, for each one of these chromosome coordinates, how many copies of the alternate allele do they have out of their 2 copies. So we have, yeah, two chromosomes, which means 2 copies of this like chromosome coordinate. So it could be 2 of the reference or 2 of the alternate allele, or 1 of each for each person, basically. Again, just to repeat something I said already, this data format is like a simplified data format. I try to make it easy on us for the educational purposes of this course. If you guys want to see what the real life like data file actually looks like for this type of analysis, I'll put a link in the description to another video that someone else made that does a good job, I think, of walking you through like the real life data format for this type of information, basically. But yeah, just for the purposes of our course, I decided to simplify it a little bit for educational purposes. So yeah, that's it for this video. In the next video, we'll start actually like writing some code to start analyzing this thing, basically. So yeah, thanks for watching, and see you guys next time.